I'm going to briefly go over development a little bit, and plasticity is the changing of synaptic strengths with some kind of experience. So when we're stimulating, the brain is always taking in information and adapting. And so when we're trying to develop a prosthetic or something like that, we have to take into account that the brain is not a static thing. It's really interacting. And it's tremendously powerful at just finding any information that it can from some signal and utilizing it. And this is something we have to take to our advantage. So I want to go through and talk about a little bit how everything is structured. So we find that the brain is constantly adapting given to its environment, and the environment can be a prosthetic that you're developing. When you lose a sensory input, like you lose an arm, there is a rapid rearrangement of the cortex, and I'll show you exactly how rapid it is. It is truly phenomenal. The brain adapts giving any kind of information coming from a prosthetic or something else, and we can also make use of that there are many signals the brain uses in development, and we can coat electrodes and things like that to promote the biocompatibility. So you can make use of developmental cues to promote biocompatibility. And then the brain adapts, so it's the same stimulus given to a naive brain. We have a different response from the brain than after you've been stimulating it for hours and days and months and years. So, we know there's over 100 billion connections, and these connections are not just haphazardly put there. They have been refined over time. There's tremendous amounts of structure in the connections. And the brain really starts out as a tube, a neural tube. So you have your blastula, and on the blastula you have some kind of thickening, and then that thickening ends up becoming a, we'll show you here. So this is on top of some large blastula. I forget the details of all this, but you get this thickening, and you get several layers of tissue. So here is a cut through, and you end up seeing 3 layers here of the developing embryo, and you get these what are called neural crests that start to form, and it ends up starting to make little bumps, and then these neural crests get taller and taller and taller, and they end up closing up and making these bumps up here and the tube. And this tube ends up becoming the central nervous system. And then you have these guys that are right next to it, which become the peripheral nervous system. And so that tube ends up becoming the ventricles and the cerebral spinal cord. The canal, the spinal cord. So there are multiple layers of this. There's the endoderm, the mesoderm, and the ectoderm. And it turns out that the central nervous system really comes from the ectoderm. And the ectoderm you will often see in developmental abnormalities like spina bifida. There'll be like a hairy patch over that area, and that's because the skin and the nervous system are really coming from the same part in development, and any abnormalities in the spinal cord often will show up in abnormalities on the skin as well. So here is, I think we've seen this picture before, but here is this tube that's forming, and at the top of the tube you have these sort of 3 bulbs around these ventricles, and these end up becoming everything around this bulb becomes the forebrain. This all becomes the midbrain, and then here's the rhombencephalon or the hindbrain, where you get this is where you see all those nuclei like the locus coeruleus, and down here is where you're getting the medulla oblongata, and then down below it is the spinal cord. So when you look at the edge here, it starts out as a little tube, but all the cells start forming right around the edge of this tube. So you have, those are the ventricles, and there's tissue right next to the ventricles called the ventricular zone. And this is right at the ventricles is where all the cells are formed. And the cells start at the ventricle, and they actually migrate out, and then migrate back up to the pia surface, and they migrate back. And then once they've migrated back, then they actually divide. So this cell here is a progenitor cell. It can really become any kind of neuron in the brain. And then this guy, when it divides, one of them continues to be a progenitor cell, can become any kind of cell, a stem cell. And the other one really becomes differentiated and just becomes a cell that is identified by the layer that's being formed at that time. So this is the layer of the brain that's being formed, and these are where all the cells are dividing. And they can split in two different ways, vertical and horizontal. This guy will stay differentiated. These guys will continue going up and down. So here's an example. This is early on. You have the ventricular zone here, and then you have these neurons that go up and into the subplate, and then here's the cortex, and then they will form some cells here, and they will keep forming layers on top. These two layers, the cortical plate and the marginal plate, will keep being pushed out, and you get a layer that forms between the subplate, and then the cells will migrate up. Once this layer 6 has been formed and go up, start to identify them as layer 5, come back down, split, come back up, and for layer 5. And once layer 5 is formed, they will continue going back down and up and form the subsequent layers. So it's formed inside out. And there are diseases where you will find the layers get formed inside out, and people will have cortical warts and associated with all sorts of bad developmental problems.